and welcome back to the Global Agriculture Innovation Forum. I'm Gerald Shively, Associate Dean and Director of International Programs in the College of Agriculture at Purdue University, your host for this year-long series of events in which we are exploring the frontier of agricultural innovation. I'd like to thank USDA, especially the Foreign Agricultural Service, for its support of forum events. On behalf of the organizing committee, our advisory group, and everyone contributing to this effort, welcome you. Today's session focuses on genetic innovations in livestock and fisheries. In case you were unable to join us for past events in the series, including the previous session on genetic innovations in crops, I encourage you to visit the forum website where you can find links to recordings of previous events along with links to a variety of other resources tied to session topics. And now, without further delay, I'd like to turn things over to our moderator for today's session, Dr. Adebola Adesogan, Professor of Ruminant Nutrition and the Director of Food Systems Institute and the Feed of the Future Innovation Lab for Livestock Systems at the University of Florida. Please enjoy the program, and please stay for the live question and answer session that will follow the presentations.
Thank you very much for passing on to me. Good morning, good afternoon, good evening, ladies and gentlemen, from wherever you are calling. I want to especially thank Purdue University and USDA for the opportunity to moderate this session. My name is Bola Adeshogun, and I'm a professor of animal science and director of the Food Systems Institute and the Feed the Future Innovation Lab for Livestock Systems at the University of Florida. Today we have a session on genetic innovation in livestock and fisheries. And this is part of the Global Agricultural Innovation Forum, which is jointly supported by Purdue University and USDA. They just concluded a session on genetic innovation on crops, so please look at the forum website for more information. The topic of genetic innovation in livestock and fisheries is very important because of the low productivity of local breeds of livestock. Many of them are adapted to their local environments, but whether it's meat or milk or egg production, the levels of productivity are a fraction of what is possible with improved breeds. And if we think about the US, genetic innovations as well as others have resulted in huge advances. Consequently, compared to 1944, if we compare the data from 2009 to 1944, in 2009, 21% of the animals that were involved in 1944 are producing the milk. 23% of the feedstuffs are required, 35% of the water, and only 10% of the land is required to produce the same 1 billion kilograms of milk. That is from the study by Kapper and Bowman and colleagues in 2009. And when we think about greenhouse gas emissions, again, the modern systems are producing 24% of the manure, 56% of the nitrous oxides, 43% of the methane compared to what they produced historically. A lot of this is because of genetic improvements. And for instance, in 44, 37% of the herd in the US was made up of Holsteins, but it's now about 90%. And that coupled with adoption of artificial insemination and genetically superior animal selection for those has really advanced the whole industry. So this is a really important area. And today we have lined up some really high caliber speakers that will be addressing us on innovations in poultry, innovations in livestock, and innovations in aquaculture. So first of all, for genetic innovations to increase nutritional quality, we are going to be listening to Dr. Susan Lamont. And she's the Charles F. Curtis Distinguished Professor at Iowa State University. Her research has focused on elucidating the genomic control of important biological traits in chickens including resistance to disease and heat stress. She also focuses on the impact of functional genomics on biological performance. She's a fellow of the Poultry Science Association and the American Association for the Advancement of Science, and she's been awarded the Merck Award for Achievement in Poultry Science. Second, we will listen to a talk on genetic innovations to increase productivity from Dr. Steve Kemp, who is the program lead livestock genetics program at the International Livestock Research Institute. And his research focuses on informatics systems and genomics of tropical adaptation, particularly host pathogen interactions and mechanisms of tolerance and resistance. He's a professor in tropical genetics at the University of Edinburgh. And finally, we'll listen to a talk on genetic innovations to enhance resilience to biotic stress. And this is from Dr. Rose Basita, who is a global scientist at World Fish. She is based in Zambia, and her research in fish genetics is helping to advance sustainable aquaculture production in the Southern and Eastern Africa region. She leads the genetic improvement program of Oreochromis andersoni on the wider Zambia Aquaculture Development Project within the government of Zambia. So with that, I'd like to invite Dr. Lamont to give her presentation, Genetic Innovations to Increase Nutritional Quality. Thank you.
Thank you for that kind introduction. Good morning, good afternoon, good evening, esteemed guests all around the world. I'm very happy to be here with you today to talk with you about genomic innovations in poultry. I'll start out by saying, why are poultry important for food security and food safety around the world? Then we'll talk about how to approach genomic research and the applications of that research with some success stories. At the end, I'd like to speculate about some future applications in poultry genomics for food security. The goal of the genetic innovations that are applied in poultry are to produce healthy, productive, and climate-resilient birds. Why are we interested in using animals in the context of global food security? They're portable. They can be moved. In case of conflict in an area or climate disasters, the animals can be moved as populations move to different areas. Importantly, they may be providing draft power on the small farms. They're also a very important part of a sustainable agricultural ecosystem. Animals can be considered a living bank account. It can be used to meet unanticipated or planned needs, such as school fees or medical expenses. And as we see population growth and increased income, there is an increase in interest for people to be deriving part of their protein sources from animal-derived sources. So animals fill a very unique niche in terms of food production. Specifically, why does poultry have an important role? Poultry, being small, are very accessible to smallholder farmers. They have minimal space requirements and a rapid generation interval. Importantly, they have very frequently available product in the form of a small bird that can be harvested for a family dinner or feast. And the eggs are frequently available in a convenient portion size for incorporating into the meal. They have very wide cultural acceptability, and in many areas, they are managed specifically by women and by youth. So poultry are very important in terms of food security in developing countries. Poultry are also very climate smart. On this graph, where we see the energy efficiency of conversion of calories from feed into the animal itself, we see that the 2 out of the top 3, eggs and poultry. Whole milk is the only one that is more efficient in terms of meat production on the land. So they're very climate smart. And why are eggs important? They're a very fundamental source of protein and essential nutrients, having many micronutrients that are absolutely essential for brain and cognitive development. So that first 1,000 days of life is very well supported by incorporating eggs into the diet. And they're also a very highly valuable trade commodity. So what about genomics? Why is genomics a good approach? It can be used to enhance the health and efficiency of the birds. It's an environmentally sound approach. The improvements that are made are sustainable. They're gradual and they're permanent. They accumulate over time as the population is improved. And we know that animal health is also related to human health. So the interest in increasing disease resistance in birds also translates to a better environment for humans. Genomics is very timely. These molecular technologies are now available. So we have the opportunity to integrate genomics and biology in a holistic approach. Because remember, the chicken was the very first of all the farm animal species that was sequenced. How do we approach finding the correct genetic selection criteria? In general, these studies will approach it in this way. We will challenge in a controlled condition the birds with either an environmental challenge, such as heat, or perhaps exposure to a pathogen, and measure their response at the same time when we measure the genetic variation. And then with bioinformatic analyses, these two types of data can be incorporated to discover the genomics that control these important traits. Different genetic selection tools are going to be appropriate for different approaches. We might be able to apply whole genome studies and applications in places where there is adequate financial backing to do so. In other contexts, we are going to have a more targeted approach of single genes or biomarkers that can be more economically and easily measured. Why would we even consider working with different genetic populations in their applications? Well, commercial lines can have value if there is the possibility to have very refined inputs in controlled environments. They are very efficient and can have rapid translation into use. They would be suitable for the mid to large scale. And in these types of applications, the very large scale types of genomic assessment are done and applied. This is already being used frequently by all of the large commercial poultry genetics companies. However, it's important to consider the value of local ecotypes. Local ecotypes are very important. They're adapted to the local environment. They're culturally relevant, and they may be very suitable for smallholders. So it's important in terms of the application of genomics to be able to meet the diverse needs of the final users around the globe. I'll give a couple of success stories from the work that I've been engaged with in the international arena. The first one is part of a U.S. Agency for International Development project, where we are looking at some of the stressors that local ecotypes of birds encounter. Newcastle disease being one of the most important disease constraints for poultry production in many of the developing countries. And heat stress also is a very important negative impact on poultry production. So we can use genomics to do a very important first step, and that is to characterize local biodiversity. In this very colorful diagram, what you see are the scans of the genome of 3 populations from 2 different countries. On the left-hand side, 3 different populations collected in Ghana, and on the right-hand side, 3 that were collected in Tanzania. And you can see how the power of genomics allows us to readily distinguish the different countries of origin of these populations. And by the similarities of colors of the adjacent blocks, we can see which populations are closely related to each other, even though they may have been collected at quite distant locations within that country. And so we are able to characterize in a very fine manner the genetics of these birds, and that's the starting point to understand how to best utilize and how to improve them. In another example, we can see how genomic variation was identified that was associated with meat yield under heat stress. In the picture in the upper left, we can see from the same age the harvested breast muscle from a typical commercial broiler and a local type of indigenous bird. And you can see the great difference in size that is inherent there. When we conducted a genomic scan on a cross of these populations, that allowed us to identify that there is one specific region in the genome of the bird that controls the amount of breast yield that is produced in those birds. Very, very importantly, what we found was that the beneficial genetic variation came from the non-commercial line. It came from the local ecotype. So even though those birds, when compared to broilers, generally yield less breast muscle weight, if they are under the stressful conditions that are quite common in developing countries of heat stress, they contribute important genetic variation that allows them to grow well and to have a good yield. So this points out the value of the local ecotypes and their adaptations to their environments and how that is important to consider in using genomic applications. So genetic innovation impacts can be at least threefold. We can develop practical tools to use the existing biodiversity in populations for genetic improvement. We also get an enhanced fundamental understanding of the biology of these animals, and that is useful also for improvement. And this will address the goal of improved poultry health and welfare, production efficiency, pre-harvest food safety, and global food security. So for just a minute, I'm going to put on my fortune teller's hat and try to speculate on what I think will be some of the future applications of genomics in poultry. Certainly, addressing climate resilience, pathogen resistance, and vaccine readiness are going to be three of the traits which are going to be improved by applying genetic innovations. Another area will be early identification of sex of the birds. Now, those of you that might work primarily with mammals might question this goal, but let me assure you that it is not easy to tell the difference on day-old chicks between males and females unless they have had specific genes crossed into that population. And because it allows us to produce the birds more efficiently, if we can identify the difference between males and females early, this is a goal for genetic applications. Studying the microbiome, which would be the genetics of everything that lives inside the bird, is also going to be important for us to improve feed efficiency and health, as well as the example that I showed you of being able to characterize and therefore preserve biodiversity within a population, especially globally as we move to looking at new populations and how they may be useful for food security and food production.
Thank you very much, Dr. Lamont, for that very insightful presentation, all the way from the importance of poultry in the diet and in livelihoods, talking about specific genetic innovations, including exploiting local ecotypes, talking about future or genetic areas of emphasis. So really, thank you a lot for that presentation. And I want to remind participants to post their questions in the chat. We'll be taking those at the end of all three presentations. We'll have a question and answer session. Now I'd like to turn it over to Dr. Kemp, who will be presenting on genetic innovations in livestock.
So I'd like to take a few minutes and talk about the potential of genetic innovation to improve the lives and livelihoods of smallholder farmers, particularly in developing world contexts. What is the potential of genetic innovation to sustainably enhance the livelihoods of farmers in developing countries? And I particularly stress that word sustainably right at the beginning, and it's a thread that will be running through this presentation. We need to ensure that it's sustainable in both a planetary sense and also sustainable in a human sense. So I want to give you a brief context, and then I want to ask, what is it that we want to achieve? and how can we use that to guide our efforts to prioritize where we're putting our work? And I'll give you an example of a genetic innovation, and then come back to this theme of ensuring sustainability and ensuring we're doing the right thing. We're genuinely responding to demand. So first, for a bit of context, livestock are really, really important. You can measure it in many different ways, but this is a simple economic measure. Livestock is 40% of the agricultural GDP globally, and in low and middle income countries that ranges between 50 and 80%. Milk is the most important, economically important commodities, significantly more important than rice, for example. And in fact, 5 of the top 10 commodities of the most economically important commodities are animal source. And as people get richer, they tend to get more of their calories from animal proteins. And that's been shown again and again as GDPs of countries starts to tick up, then the demand for animal source products follows fairly quickly. And the reason for that is really clear. Animal source products are extremely nutritionally important. They're not simply sources of calories. They're sources of micronutrients, vitamins, and so particularly in a resource limited setting, a small amount of animal source food can be incredibly important for health and welfare. So we can anticipate an increase in demand for livestock products in developing economies. How could we meet that demand? Well, we can see three ways of doing it. We can import livestock products, we can import industrial scale livestock systems, or we can look at transforming the existing smallholder livestock systems. And I want to particularly focus on the last of those three. And it's pretty clear that smallholder farmers can meet the world's increasing demand for meat, milk, and eggs. Already, something like 50% of the world's agricultural products come from smallholder farmers, farms of less than 20 hectares. So we're quite confident that we can significantly improve the productivity of smallholder farming systems. And now I want to pause for a second and say, discuss the philosophy of how we decide what we're going to do here. It's very easy just to pile in, and especially if you've got some cool biotechnology tools to say, hey, this tool can solve, let's try this tool. Let's see if this tool works. It's really important, I think, to be systematic about this. And a friend of mine who works in the pharmaceutical industry has a really clear take on this. He says, if you want to fix something, you innovate around bottlenecks. You say, what do we want to achieve? What's stopping us from getting there? And how do we fix those bottlenecks? And I think taking an approach like this, when we come to applying biotechnology and genetic innovation, is really important. It prevents us, it ensures that we're demand driven and stops us being driven by our latest toys. And I think we can see there's actually two kinds of bottlenecks. There's pure hardcore technical bottlenecks, something that we can't do. We just don't know how to do it at the moment, or it's ridiculously slow or expensive. And you can think of any number of examples of those, those sort of magical innovations that we'd love to have. But there's another kind of bottleneck, and that's the capacity. Often we know this can be done, but it's not happening. Why is it not happening? For example, why are we not sustainably improving breed performance? Why are we losing biodiversity? And those, of course, are interrelated. They feed back into each other. So, and when we start to look at the context and ask, what is it that we want to achieve? We see that we're immediately faced with trade-offs. So obviously, we want to improve ultimately animal nutrition health in order to improve human nutrition health and food security. But that comes with a whole bunch of consequences around disease risk, unsafe foods, the risks of overconsumption associated bring associated problems of obesity and non-communicable diseases. We want to use livestock systems to reduce to reduce poverty and to improve livestock, to improve livelihoods. But it can be a real struggle to make sure we do that equitably. And the benefits remain with small scale livestock keepers, and the benefits are equitably distributed amongst men and women, old people and young people, and so on. And we also need to be very careful that what we're doing is sustainable in a planetary sense. We need to reduce greenhouse gas emissions, for example. That is a clear imperative, and we need to be able to adapt to the consequences of climate change. So, but sometimes it's a real win-win situation. And one great example here is the greenhouse gas production associated with the milk production. If you are, so what this graph shows is that as each cow produces more milk per lactation, the greenhouse consequences of each liter of milk comes crashing down as you get more efficient. So 90% of Ethiopian milk production is producing just a few hundred kilograms of milk per lactation, and a load of greenhouse gas is the cost of that milk. But the best 10% of Ethiopian milk producers are a little bit more efficient and produce much less greenhouse gas for each liter of milk. So our opportunities here, for instance, to reduce the greenhouse gas footprint of livestock is really very clear. We're working at this very steep part of the graph here, where modest improvements in performance produce dramatic improvements in sustainability. So where do we want to get to? If we look at milk production in the US, for example, this graph simply shows an absolutely relentless improvement in milk production from the 1960s up to the 2010s and beyond. And what drove that? Was genetic improvement. It really is as simple as that. We can show empirically that it was the genetics that did that. It wasn't the farming systems, it wasn't the feeds. They had to keep up with the genetics, but it was primarily driven by genetics. Now, why is that not happening in the developing world? And it's not. A graph for most parts of Africa would be flat and would show no improvement. And we can say that some of those reasons are characteristics of the system, but some are problems with the infrastructure. So I particularly look at those, at the constraints around the infrastructure. That improvement was driven by artificial insemination, data recording, and selection schemes. Why aren't we able to do those in the developing world? Or how can we innovate in order for that to be possible in the developing world? So there's huge potential, I think, for innovation around performance recording. We can skip a whole generation of technology. Performance recording in Europe and the US, for instance, is done by pretty clunky, old-fashioned recording of milk production, for instance. There's a real chance here, I think, in Africa to use modern technology, mobile technologies, low-cost sensors, crowd sensing, to gather performance data in a really novel and really lightweight way that we can use to drive genetic improvement. The other extreme bottleneck in genetic improvement, particularly in dairy systems, is genetic delivery. If I could invent the perfect cow for the whole of Africa tomorrow, I would have achieved nothing because I wouldn't be able to get it to the people that need it. The big problem is getting the right genetics to the right place at the right time. So artificial insemination is what's allowed that to happen in the West, but it's actually a really fragile system. It works very nicely in the US and Europe, but it needs an unbroken chain of liquid nitrogen. And Africa is littered with dead liquid nitrogen plants, and every office of every breed organization is full of broken liquid nitrogen containers. It's really difficult to identify estrus, and you've got to do that quickly in order to call up your artificial insemination technician to come and deliver the right semen in the right state of health within 24 hours of the farmer detecting estrus. This is a really fragile system. It depends on an extended infrastructure, and it basically doesn't work. And that failure costs farmers every day and prevents genetic improvement. There's a really clear bottleneck. What can we do about that bottleneck? I think there's some really interesting technology emerging from Washington State, and we've got a collaboration going with John Oatley, working on surrogate sire systems, whereby he's looking at making a surrogate sire that doesn't produce its own sperm, but can be populated with cultured sperm from an elite genetics that you want to distribute. So effectively, you are amplifying the bull or the buck that you want to use, and equally importantly, you're putting that system into you're putting that sperm into a bull or a buck who replaces the artificial insemination supply chain. So effectively, it's like a mobile semen flask that is very capable of detecting heat and is very capable of delivering semen. And I think this is an example of a biotechnology innovation that has potential to be really transformative in developing world systems by just blowing away that huge bottleneck associated with artificial insemination. And we can show modeling that system shows that we can expect really dramatic improvements if we were to use the surrogate sire system in genomic selection. We would expect to move to be at a particular place 6.5 to 9 years faster by using a surrogate sire approach compared to conventional sires. And that is simply as a result of amplification. We've just got more of that of the target genetics that we want, is more of it available. But of course, in the developing world, that's not the major problem. The major problem is all of those infrastructure bottlenecks that we're also blowing away by introducing the surrogate sires. And getting rid of the need for liquid nitrogen, getting rid of the need to detect heat, call up a bloke on a motorbike to come and do an insemination. So I think that this surrogate sire technology is really exciting, has real potential for transforming systems, enabling genetic improvement, and then becoming a vehicle for delivering other kinds of genetic improvements. So for instance, you might imagine using it to deliver genetically modified animals that are disease resistant, for instance, because there's no point in making a disease resistant animal if you can't distribute those genetics. And finally, I want to touch a little bit on the capacity to do biotechnology in Africa. We tend to be very supply driven as scientists and biotechnologists. We tend to sit around thinking, what's a cool thing to do? What's fun science? And then we tend to say, and now we can help poor farmers, or we can help African scientists with our wonderful ideas. And we tend to get it wrong because we don't understand the context. And there's a long history of inappropriate interventions and very short-term targeting by scientists and donors. And I think one of the really important things is to take a much longer-term perspective about the capacity to do biotechnology in LMICs, lower and middle income countries. In particular, I'm thinking here of Africa, where Africa tends to, where African science is driven by donors, and donors are by definition short-term, and they are seduced by their national scientists saying, hey, I've got this really cool thing that will just change, that will be fantastic, and kind of imposing it. So the plea I would make is that the capacity is that donors need to stop being part of the problem and start to become part of the solution. So we tend to block the development of local capacity by flying in experts. We tend not to acknowledge the depth of understanding of the system that's already present on the ground. And if we do that, I think we can make biotechnology much more demand-driven, much more context-aware, and much more sustainable and long-term. And there's a beautiful quote from a report, from a NEPAD report on the science agenda for agriculture in Africa. And I think one of their nicest quotes there is that science for agriculture in Africa is too important to be outsourced to international investors. And so I think on that note, I can finish. Thank you very much.
Thank you very much, Dr. Kemp, for a really interesting presentation, starting with the emphasis on planetary and human sustainability and how we need to ensure that we address those aspects. You mentioned that smallholder farmers already meet the global demand, 50% of the global demand for livestock products. And you talked about how trade-offs are often necessary when we think about improving productivity versus the environment. You give the example of the win-win with milk production in the West. And then you mentioned the constraints that have prevented similar benefits from potentially groundbreaking genetic technologies, the distribution issues, the issues with AI, and then surrogate sires, and how that can really be a game changer. And you finished with some interesting comments about donors. So thank you very much. Really interesting presentation. I will remind you to please post your questions in the Q&A. And we have one more presentation, and then we'll open it up for questions. So I'm going to invite Dr. Rose Basita to speak to us about genetic innovations in aquaculture and fisheries. Thank you.
My name is Rose Basita. I'm a scientist with Worldfish and the Sustainable Aquaculture. I'm based here in Lusaka, Zambia. I'm glad to speak to you this afternoon on a very important topic, genetic innovation in fisheries and aquaculture. I also take this opportunity to thank Purdue University for the opportunity for us to speak on such an important topic to the Global Agriculture Innovation Forum. Thank you very much. Genetic innovations in fisheries and aquaculture. Why fisheries and aquaculture? This is because aquatic organisms, including fish, are a good source of nutritious food. For instance, if we look at fish, it's rich in micronutrients. These range from zinc, iron, vitamin A, calcium, and it depends really on what species, but they're essential for cognitive and physical development, especially among the children. And it's also an important part of a healthy diet for any age group. Fisheries, or particularly aquaculture, has a very small environmental or carbon footprint. And this is relative to, for example, beef and cattle. And the number of studies that have proven this, studies run in Iceland, Canada, Sweden, USA. Actually, I like a study by Boyd in the US that considers pond aquaculture to be carbon neutral. So most farmed fish it's important to note that it's from fresh water. Over 96% of farmed fish is freshwater fish, and just the remainder is marine. And 50% of fish eaten globally currently is from aquaculture. For obvious reasons that the capture fisheries, the fish production has sort of plateaued for quite some time, and we see a tremendous growth in aquaculture. However, like any accelerating industry, we do have bottlenecks, and these have been largely around fish seed, feed, husbandry practices, and funding being key challenges in the sector. And very recently, because we're intensifying aquaculture, fish diseases have become paramount. So it caused a new challenge to the industry, hence the need for biosecurity. But I have some good news. Genetics does pose immense potential to accelerate the industry to meet the development goals in aquaculture. What genetic innovations exist in fisheries? There are quite a number, but it's important for us to note that they're quite recent. Genetic interventions traditionally that enhance animal and plant production have been known in the mainstream agriculture production, that's plant and animal, and it dates far back. It's only of recent, hardly 40 years, that we've seen genetic interventions in fisheries management and aquaculture. And within the aquaculture sector, the major traits that have been targeted have been growth, feed efficiency, disease resistance, and of course the focus being on growth because it's easy to measure, but also it directly translates to increased production, profitability, and hence food security. So the initial breeding programs largely focused on growth, and it's really justified. Despite the fact that a number of species have been improved, given the importance of genetic improvement for aquaculture food production, species like Atlantic salmon, shrimp, and from the tropics, tilapias, they've had a systematic genetic improvement work done on them, and this has driven their global success in terms of production. However, we have a lot of unimproved material, and a lot of the aquaculture industry depends on these aquaculture species that are not improved. And it's important to note that adoption of proven genetic improvement programs has generally been slow. According to the state of the World Aquatic Genetic Resources with the Food and Agriculture Organization, the global plan of action really calls for accelerating the appropriate development of aquatic genetic resources for aquaculture. This is because selective breeding programs could meet the future demand of fish, of course, not as standalone, but combined with inputs such as feed and good husbandry practices. And the emphasis here is developing long-term and well-managed selection programs, because a number of programs start and fail because of a lack of that initiative and initial plan to see that we need to develop planned long-term and well-managed breeding programs. I'll pick up an example as a success story in terms of genetic innovations in aquaculture, and this has been the GIFT technology in aquaculture. So GIFT denotes genetically improved farmed tilapia. GIFT was developed by Swordfish and partners. It started in the Philippines then, but currently we are doing the selection from Malaysia in Penang. The technology has widely been adopted, and the fish is farmed, the GIFT tilapia is farmed in almost the whole of Asia, or the most part of Asia, but to a smaller extent in Africa. By 2015, the distribution has reached over 16 countries across the globe. Given the advantages of genetic improvement in aquaculture, the GIFT tilapia alone has shown, impact studies have shown an increased production over non-improved strain by almost 85%. And this is calling for just the major GIFT tilapia, but also the number of GIFT derived strains. So the commercial companies have picked up the GIFT and also tried to develop. So combined, we see that they are now thought to provide over 50% of the global tilapia production. So it has been a tremendous entry point into tilapia production globally. I mentioned earlier on that the major traits that were targeted were growth and survival. And the growth rate, improvement rate has been 7 to 10% per generation. This sounds quite high compared to the livestock improvement per generation. So it's really faster in fish. What has been the recent research that we've anchored on, especially in the area of gift? Well, as WorldFish, the recent work on gift has concentrated on sort of speeding up selection, but also efficiently doing this through genomic selection. This is because with the aid of genomic selection, we are able to particularly target resilient traits in order to produce a fish that is more resilient and do it with greater efficiency. So with the use of genomic selection, we've been able to target traits like feed efficiency, disease resistance. You might not be aware, but the tilapia lake virus has been a virus that has hit the tilapia industry in Asia, both Southeast Asia and the Middle East, talk of Israel. So it's been a menace, and part of the gift targets presently is to target disease-resistant traits as one of the key avenues for in genetics. Another key trait that we're looking at is adaptation to variable oxygen levels. We know that most tilapia is farmed by smallholder farmers who do not have the equipment to have aerated ponds and things like that. So targeting a trait like adaptation to variable oxygen levels is paramount. And this has been part of the target in the last couple of years with the gift tilapia. Further to the development and research, we see the tools that have been developed, molecular markers, and one of the key successes that we've had is having an open access tilapia SNP chip. And the gift genome sequence has now been developed as well. Of course, similar trends are being seen in the other species like the salmon, the shrimp, and like the tiger prawn, salmon, and other fish industries. So it's not just unique to the gift tilapia. And it's true as well for livestock. The inclusion of resilient traits like reproduction, disease resistance, immune competence have just been achieved because of the availability of genomics. And aquaculture has also taken that window of opportunity. We see an opportunity for improved strains using molecular markers, and this has been exemplified with a number of countries coming to us, for example, in WorldFish, governments wanting to start up breeding programs. There is a new one we are starting here in Zambia with the 3-spot under Sunai. In Malawi, we have the Shiranas. But we are seeing more and more hunger to have genetic improvement programs at government level, but also the private sector and the industry embracing the technology of selective breeding in fisheries and aquaculture. So integrating genomics in breeding program is really the way to go because you can tackle a number of traits concurrently. So what have been the current and where do we see the industry going in the area of genetic interventions and innovations? We see more use of genomic tools in fisheries management, especially traceability, because genomic tools have allowed us to, with a far greater sensitivity and accuracy, we see an increased precision having turned out in aquaculture species. This is also true with the capture fisheries. It gives us cheaper surveillance and rapid extensions to like what you would say, the novel species, we can extend it to new species that we're not traditionally having, like they're not commercial to the extent that they have like SNP chips developed for them specifically, but we can go with the genomics and the low genotyping costs that are coming along on the market, we'll be able to extend to more species. The high potential to detect diseases and monitor wild stocks as well as farm stocks will only be possible with the use of genomic tools. Selecting for disease-tolerant traits for climate resilience is also going to be paramount as well. We look ahead. Yeah. One other example of DNA techniques I thought I would also share is the issue of environmental DNA. Instead of sampling tissues directly from fish for analysis. It's to tackle the water environment where the fish are. This helps us understand the presence of species, both in the wild, especially in the wild for capture fisheries, the community structure. We've used eDNA, for example, in a number of water streams in the US for detecting invasive species like tilapia is not native in Australia, and we use the eDNA techniques to identify places before even other methods that have been used traditionally, like electrofishing, have been used. So it's a non-invasive way to detect invasive species. You can use it also to detect presence of pathogens, for example, in ponds, before the disease becomes a problem to the crop. So such detective methods like eDNA are going to be very important in fisheries management presently, but also in the future. I've already mentioned about detection of invasive species. What can we take home from this talk? We see that improving production in aquaculture is the core for selective breeding programs. And just as is the case with agriculture, and we've seen this exemplified with the salmon industry, and I've elaborated in this particular talk, the tilapia industry, but this is also true in the prawn or shrimp industry. Of course, the key trend is the inclusion of more resilient traits beyond growth that has been studied previously to include resilient traits and the use of index selection. The use of genomic selection for improved accuracy and speed of selection. And again, as I concluded, we see the use of molecular and DNA tools enables developments in aquaculture for better management of wild stocks, including the interaction between the wild and the cultured stocks, because they go hand in hand. Thank you so much for listening to me, and it's been a pleasure talking to you.
Thank you, Dr. Basita. Thank you very much for that insightful presentation, starting with highlighting the nutrient density of fish and importance in a healthy diet, the relatively smaller footprint of fish in aquaculture, the importance, or a growing importance, already 50% of fish consumed is aquaculture, and then the rate of increase is very, very high relative to farmed capture fisheries, relative to wild fisheries. Then you talked about challenges, productivity, low productivity, fish seed, fish feed and diseases, low adoption of technologies, and then you shared about the gift tilapia and how that can increase the productivity. The growth rate is 85% greater, and it's taking over about 50% of the tilapia market. And you elaborated on some of the specific traits that were selected to get that gift tilapia. And you finished with some of the future areas, traceability, cheaper surveillance, more precision, disease detection, and so on, that you can achieve through genetic selection. So thank you very much. So please continue to post your questions in the Q&A session. What we have heard from our three speakers now, and it is very clear from what they've shared, that very important improvements in growth, efficiency, reduction in emissions, improvements in health, improvements in livestock product quality can be achieved through genetics. So I'm going to start with a few questions for each speaker, then we'll go to some questions that we'll throw out to all the speakers. So Dr. Lamont, let me start with you. You talked about the effect of heat stress on poultry productivity and selecting for heat-tolerant breeds. To what extent are genetic solutions being implemented in this area, and how successful are these?
Thank you very much for that question. I feel that heat stress and resilience in the face of environmental Stressors is certainly one of our limiting factors in continuing to develop animal production efficiently around the world. Some of the ways in which improvements are being made in poultry in heat stress resilience are closely related to genetics in that they are addressing things such as manipulating the microbiome, and that would be via choosing to feed animals the types of things such as probiotics that would help them to have a beneficial microbiome and therefore be able to maintain a good level of feed efficiency even under heat stress conditions. So that is related to genomics, but it's really altering the genomics or populations of the microbiome that is within the gut of the animal. Another approach that is being taken is to use thermal conditioning. And thermal conditioning is thought to act through what is called epigenetics. So not changing the genome itself, but rather changing the way that the genetics of the animal is expressed in its physiological traits. And this thermotolerance treatment might be applied either on the developing egg itself or on the newly hatched chicks, with the theory being that exposing the developing bird or the very young bird to an episode of heat allows the genome of the bird to have certain changes that will allow it to then better respond when it later in life encounters an episode of heat. So those are a couple of the approaches that are being taken. Another one is certainly the simple utilization of animals that have a historical development within areas of the globe that have a very high heat load as a part of that natural development of those populations and using them as the resource. So 3 different areas: the microbiome manipulation, thermotolerance treatments, thermal conditioning, I should say, or actually using those populations whose historical development has been in those parts of the globe. Thank you.
Thank you, Dr. Lamont. Dr. Kemp, you said you need to get the right genetics to the right place at the same time. So the surrogate sire system is very intriguing. To what extent can this be scaled not only across geographies, but across species and systems?
Yeah, thanks. It can certainly be scaled across mammalian species. So we're currently working in a goat model, but John Oatley in Washington State has already demonstrated this in cattle and in pigs, because it's the same gene. Knocking out this gene called nanos2 completely ablates sperm production in the host animal, and that's really all you need to achieve in order to get a surrogate sire. So across species, absolutely, yes. And in fact, it's in small ruminants that we think there's greatest opportunity, because there are not currently systems for artificial insemination. Genetic gains tend to be driven at a very community level rather than nationally or transnationally. And so we think that the surrogate sire system can allow, communities are already sharing the right animal, but we think the surrogate sire system can make it possible to share, the ideal buck or the ideal ram more widely and have its genetics more widely available. And indeed across geographies and across systems. I'm not sure what you mean by across systems. There are some I can imagine some systems where it wouldn't help very much. Where artificial insemination is already very well established, surrogate sires don't bring as much to the game. But across geographies, absolutely, it's a relatively simple method. Once you've got your recipient ready to receive the genetics that you want to disseminate. It's just a question of identifying the elite animal that you want, deciding what's the right genetics to get into a particular system, putting them into a surrogate sire, and then getting that out into the system. So it's certainly scalable across geographies as well.
Thank you. Dr. Basita, there's a question for you which says it's interesting that GIFT is widely adopted in Asia, but not Africa. And so can you explain this phenomenon, please?
Sure, thanks very much, Ebola. True, the gifts, most of the germplasm for the founder stock was sourced from Africa, actually, to try and develop the gifts in Asia. But I think initially there were concerns of having that chained fish, for example, back into the African context. But that has been evaluated over time. And we see for any responsible aquaculture, any fish species that has been improved will definitely be different from the original. So it's about adopting, again, responsible forms of aquaculture in terms of biosecurity, in terms of retention, working through systems that protect the natural environment from the farmed species and be able to separate the two. That said, we see again the gift technology being scaled in Africa by applying the technology of improvement to other indigenous species within Africa. I'll give an example here in Zambia. We've started the genetic improvement program for the 3-spot under Sonai. And basically we are using the gift technological improvement process to try and now do adaptively with the local species. So every government will be different in terms of accepting a new, so to say, gift. And depending on what resources that are already available, those countries will adopt or accept these resources differently. And we see that across Africa. It's different in Nigeria, it is different in Kenya, in Uganda, in Southern Africa. So again, it's coming back to the impact assessment studies that have been done to suit the different environments where fish should be reintroduced or not introduced.
Thank you very much.
Yeah.
Dr. Lamont, can I ask you about to what extent has the variability in productivity of local breeds been exploited? And that's the first question. And the second one is when you talked about future areas of focus for genetic innovation, you didn't mention taste. And in my work in Ethiopia, in Nigeria, and some other places, people like the broiler because of the extra meat and so on, but they don't like the taste. And the Neuler breed was, developed to try and address both the growth and the taste. So could you please comment on that too?
Thank you. I'll start with that second question, please, because it's a very important one, and it addresses the broad issue of acceptability of the product and desirability of what is being produced. And that, I think, is an essential consideration whenever we are looking at animal improvement, and that is whether there is the appropriate combination of all of the characteristics that the local user is desiring. And that can certainly be one of the reasons to include some of the local ecotypes in the development or improvement, because they do have those desirable characteristics that people are very used to having, they desire to have in what they are going to be preparing for food. It suits well for the food preparation methods. So that's one of the reasons that in some areas we see the use either of improved local ecotypes or sometimes crosses of local ecotypes with improved types of lines in order to boost some productivity and yet keep in those populations some of those extremely desirable characteristics for use of those birds. And I apologize, I'll have to ask you to repeat the first of the questions because I got so excited about your second one. I wanted to jump in on that.
No problem. First one was about to what extent have we truly characterized and exploited the variability in local breeds, leveraged,
Okay. That's a great question. I'd say that we are really only at the beginning of that journey because we don't have enough data, either the general characterization of the physiology and the performance of many of the local types of birds or other livestock that are produced. And we're just beginning to apply molecular characterization. And that's why I wanted to show that one example, because we sampled in each country that we were working in Africa 3 populations that we expected would be distinct from each other based on sampling them in geographically distant areas. But as molecular analysis was applied, it became very clear that 2 of the populations that we sampled within the countries were quite closely related to each other. And so that type of information helps us know whether or not we really have close relatedness or quite distinct populations. And that helps us know where we might identify and capture and then utilize some of the best genetic variation for use in different local regions.
Thank you.
You're welcome.
Dr. Kemp, there's a question for you here. And the person says to increase sustainability and reduce environmental impact of livestock agriculture, perhaps the best approach is to increase production. Your example of methane production per liter of milk produced, the reduction over time shows this very clearly. So should the focus be on genetic solutions rather than increasing inputs to increase production?
Yeah, that's a good question. I think it's about matching the genetics to the system. We kind of think of it as ratcheting up the system. You've got to improve the genetics, and if you do that, you've got to improve the feed. The laws of physics say you've got to put more in if you want to get more out, and then that enables you. Then you're in a system that generates more cash for the farmer. That enables you to improve management systems. So we think of it as a sort of repeated cycle, but with none of those components going too far ahead of the other. There's no point in putting in really wonderful genetics if you can't get enough feed into the animal. And no matter how good the genetics are, if the animal is half starved or is missing some key nutrients, then it's still going to be very inefficient, especially in greenhouse gas production terms. So for me, the key is matching all of those together and incrementing the genetics. I mean, I think of genetics as the driver of improvement, but you've got to follow it with improved systems and improved feed. But maybe one point is that there's a lot of discussion about genetics per se to reduce greenhouse gas production. So all other things being equal, can we breed for reduced greenhouse gas production? And I think that's an interesting topic, and it's something that should be looked at. But my instinct is that if you remember that curve showing the way greenhouse gas production plummets as you improve performance, is that the quick wins are to be made just by improving the health and the performance and the productivity of animals. And it's not until you start to move along to that right-hand half of the curve that you then begin to get opportunities to breed for reduced greenhouse gas production per se.
Thank you. Dr. Basita, there's a question for you here. It says, not all tilapia strains taste the same. Does the current gift program consider this as an important quality trait? Has there been any study on consumer preference? There could be a trade-off between more efficient, faster growth and inferior cooking and eating quality. And the person talks about the tilapia fillet they can get from a US supermarket that I will not name, and says it's very bad in taste and texture compared to something they can get from Lake Taal in the Philippines. So please, over to you.
Thank you very much. I think there are two sides to this question. It's the idea of taste. So as we progress with the use of genomics, we're able to target more traits. Initially, when we started the GIFT program, the issue was to target for growth, that is increased weight, and for reasons that I did mention in my talk, that includes increased productivity as well as profitability. That was a key issue then, because the GIFT came in to address a problem that was inferior growth and decreased production over time. But whether the fillet he gets from this supermarket is a gift is another question. So, and the issue of traceability now becomes an issue. So genetics can help us identify and be able to trace actually products that are on the market to see whether what they're saying is what it is. That said, I think it's something that we do get on board, but there's also to address the issue of hearsay rather than what it is. And I think genomics tools or genetics provide that avenue to be able to trace items that are so-called gift or not gift or whatever product they're saying they are. So the current, for example, SNP chip that has been developed is a very good example to use and trace products to clearly say, are they what they claim to be? So I am not the expert to know which particular tilapia was got there and how it tastes. But that said, it's an issue of whenever we do improvement, the issue of clients or consumer preference becomes a big issue in production. You answer one question, you get into another one. But we do have that at heart and make sure that we'll improve for a taste.
Thank you. There's one more question for you before we go into the general questions. Are there training and support opportunities for communities that may be interested in starting fish farming and taking advantage of the improvements you mentioned? And the person said, particularly in a country like Lesotho, where this doesn't seem to be present.
Oh, yes, I think I'll speak as a world fish scientist. We do have offices, for example, scattered all over. So in Africa, we do have an office in Egypt, and Egypt has a training center. So there we are doing genetic improvement for the Abasa strain. And a number of countries have benefited from this center as a training center for all kinds of aquaculture practices, including breeding, hatchery management, feeds, and things like that. So yes, we do have training programs and facilities to aid people to come and do their trainings. So a person from Lesotho, we do have, for example, Abasa. We are based here in Zambia. We do not have an office in Lesotho, but we do have an office in Zambia. We do have an office in Malawi. And this is to cater for certain needs. So I suggest that they can get in touch with us to have some recommendations. We do offer technical assistance, I think, to South Africa as well. But I'm not aware of an office in Lesotho. Sorry about that. Yeah.
Thank you. So now for the general questions, and each of our speakers can share their own views. The first one is the direction in livestock biotechnology towards, and that includes aquaculture, Dr. Basita. Is it towards genetically modified or genetically edited non-GM animals? So Dr. Lamont, let's start with you, please.
Yeah, thank you for that question. First out, I will share a personal opinion, and that is that it is essential that consumers have the information about how their food was produced. And this is a general philosophy that I hold very strongly, that consumers have the information about the production of the animals. at this stage in time, we know that we are moving towards biotechnologies that are much more refined than anything we had available in the past. And so it is now quite possible to be very specific about improvements in genetics that are being made with, for example, gene editing. But again, it needs to be done in a manner that is acceptable to the specific consuming public. And in some cases, it will be decided by opinion. In other cases, perhaps by regulation locally in the country or region environment about what is viewed to be acceptable. I think there is no doubt that as a research tool for better understanding that may have spin-offs that are not themselves genetic editing or GMOs will also aid us in genetic improvements, even if the populations that are available to producers and consumers are not themselves genetically edited. Thank you.
Thank you, Dr. Lamont. You've actually touched on two additional related questions. One is developing countries may not be ready to regulate GM animals, and this can create significant obstacles. The second one is, did you ask the producer and consumer if they're interested in these genetically edited or modified animals? So we'll continue moving with two other speakers, Dr. Kemp and Dr. Basita. Please share your views on these and the initial question about whether we should be going towards GMOs or non-GM edited animals. Dr. Kemp, please.
Yeah. So I think there's a definite move towards genome editing as opposed to genetic modification, just because there's that much more precision associated with it, and because it's regulatorily, is that a word? Regulatorily easier to do. But I would say that genetic gains, particularly in the developing worlds, it's not clear to me how important genome editing is going to be compared to just improving the system, just making genetic gains by getting your breeding right, getting your breed recording right, and doing long-term planning to make sure, to touch on a question that was mentioned earlier, to make sure we're taking full advantage of local adaptation and improving our system. So I do think genome editing will have a role to play. But I suspect it's a relatively minor role compared to some major wins that could be made conventionally.
Thank you. And Dr. Basita?
I'll not differ much from the previous speaker, but I see, for example, a role of genome editing from an aquaculture perspective, because you can use it, for example, to achieve sterility in production animals. So we see the environment of fish, for example, if you have it in an aquaculture setting and their escapees are going back to the wild. So if you can use, for example, technologies like gene editing to achieve sterility, that could be an area you could use. But for food production, we still see the conventional methods of selective breeding having greater advantages as at now.
Thank you. Thank you all for those answers. There's also an interesting question here on how can molecular characterization of animals help in the conservation of genetic resources of native breeds? Dr. Lamont, please.
Yes, thank you very much for that question. I think that the molecular characterization is really an essential part of conservation efforts because it gives us a greater level of understanding of what the genetic diversity actually is in any one specific population. It's very easy to look at a population and judge by visual characteristics whether animals are similar or different from each other. But often we find that the answer about their relatedness or their uniqueness is much more clear if we can get the additional information of looking at the genome, looking at their molecular characterization, and determining from that, with the greater level of precision that comes with that type of analysis, can help to guide conservation decisions to be sure that genetic diversity is not lost.
Thank you. Dr. Kemp, I'm going to modify the question a little bit for you, because you finished your presentation by talking about how changes are needed even with our donors. So my question, first of all, is that is this area of conservation of genetic resources of native breeds, is that attractive to donors? And then can you elaborate on some of the changes in perspective, approach, and so on that you think that the donors need to adopt?
Wow, how long have you got? There's some interesting questions rolled in together there. So, I mean, I think. Do we need to characterize, genetically characterize animals in order to conserve them? Yes, absolutely we do. And I think it's useful to contrast with the crops people. They can literally throw their seeds into jam jars and sit and wait. And then when a new trait is required, a new pathogen comes along, they can bring them out and test them. For the animal people, that's not really an option because it's so much slower and more expensive to phenotype. And so I see gene banks, animal gene banks, as being really a process of conservation combined with characterization, continually improving our understanding of what it is that we have stashed away. And in fact, you could even imagine, if we get good enough, you could imagine an in silico gene bank. You know, if we can adjust a genome at will, then in fact maybe we don't even need a physical copy of the animal. Maybe we can engineer it into using what we know in silico. But so then to go on to the question of how important is this to donors, that's a good question. You know, I think donors are responding to national demand here. Donors have not until recently paid a lot of attention to it because it's not very attractive. It doesn't show quick wins. It doesn't show, you know, how many farmers have I pulled out of poverty this week. It's about building long-term infrastructure. And we've had good collaborations with the African Union, for instance, AUIBAR. They're very aware of this, of the importance of the national resource that's represented by local ecotypes. And the donor community, I think, is beginning to come around and accept that. I suspect it's largely because of pressure from African governments. And I would encourage African governments to keep on pushing it is important. And again, it requires long-term investment. It's no good. A 3-year grant to build a biorepository is completely useless if there's no money to keep it going or no money to characterize what goes in there. So it does need a change of thinking.
Thank you. Dr. Basita, I'd like to go back to one of the earlier questions, and that's about consumer accepted of genetically modified or edited animals. And what are your thoughts about this? And what would you be saying to an audience that is skeptical?
Right. I think, again, it comes about understanding what they are. So I've had a lot of misconception, even just by the terminologies themselves. So the moment you just mentioned genetics, it's GMO, it's gene editing, it's all a mix of things. So I've found a number of audiences, even just understanding the terminologies is a problem. So I think that's a starting point, awareness raising, understanding what's going on, and then the consumers providing their input in terms of what they want out of, for example, a genetic improvement program or or whatever production aspect that we are tackling. I think it goes back to consumer rights, consumer needs, and what resources do we have available. I'm actually reminded when you asked me about the tastes and so on, we did run a study, I think last year, and we are looking at the traits that the consumers do require here in Zambia, even just before we commence the breeding program. So they are quite pertinent to us as researchers, just as they are to the consumers, and we should pay clear attention to what they need. I think that's what I can say, and it's their right to have knowledge and understanding of what's going on.
And just a follow-up question for you. The gift tilapia, how different, how similar, you talked about different strains. Is there a risk that these are genetically similar and we may have a disease in the future that wipes out vast numbers of these?
That's a good question. So when it comes to the tilapias, they are quite close. And I think I wanted to hint on the use of molecular characterization, which is very key in the aquaculture industry, where, for example, consumers or even government, without good experience, they might not be able to tell apart species or strains. But talking about a disease, I think the tilapia industry has been hit with the tilapia lake virus, and that's a key disease that has attacked the tilapia industry. And I can assure you right now, it's one of the key issues that we are looking at, even within the gift, to have resistant breeds. Even for the new breeding programs for other tilapias, it's become very paramount for us to have disease-free start founder populations, but more than that, also select for resilience and resistance to disease. So it's a key aspect. We've already had impacts from such diseases, even in aquaculture. Yeah. Thank you.
My last question is going to be for Dr. Lamont. And Steve, Dr. Kemp alluded to the importance of some of the other pillars of livestock production. He talked about feed a little bit, but I'll give you one specific example. I was called by a vice chancellor, president of a university in somewhere in Africa, and he told me, Bola, I want to bring Holstein cows. I want to bring 100 of them into our university herd because we want to increase the productivity. And he just wanted to bring the cows in without thinking about anything else, feeding, management, health. And so, Dr. Lamont, would you speak, please, about how these genetic innovations need to be embedded in or introduced with proper attention to these other factors, please?
Very much. And I think, each of the speakers have also addressed the topic partially in their talks, in that we need the good match of the support that we are going to be providing for the genetics of the animals with the environment in which they're going to be raised. And that is, as animals are improved to be higher productive animals, more efficient animals, that often comes along with the cost to provide them with better environmental control or feedstuffs, for example. So we need to be thinking about the animal production as a complete package. Providing improved genetics alone is very seldom going to be a solution, and it can be a disaster if they are not well matched for the inputs that can be locally provided for them. And that also includes the human resource development. As more refined and genetically improved animals are available, the caretakers of those animals also need to be provided with the information and skill development that'll help support the production of those animals. So I see that as an essential part of what needs to be in that complete package for improving animal production, with part of that being genetic innovation, but not all of it. Thank you.
Thank you, Dr. Lamont, and thank you to each of our speakers for really insightful presentations, great answers to questions. Special thanks to Purdue University and USDA for hosting this symposium. I think we can clearly see that there are genetic tools out there with tremendous potential to improve livestock productivity, health, mitigate emissions, but these have to be introduced in a participatory manner with scientists in the developing countries. We need to exploit local ecotypes. We need to think about sustainability, and we need to bring along the consumer. So with that, I'm going to draw this session to a close. The next session is on innovations in biological amendments. That's on June 1, so please look at the forum website for this. And thanks once more to Purdue University and USDA for hosting this symposium and to our speakers. Thank you.